Morning, everybody. Thanks for having me. This is my first Hunter's Hope conference. I'm excited to be here and talk about ALSP, adult onset disorder. So I'm starting with my summary, actually. So we'll end here, but to kind of see in my experience that I'll talk about later for the adult leukodystrophy clinic, just what needs to happen along the way to get to a disease-modifying therapy for an adult leukodystrophy. We're actually doing pretty good for ALSP. There's been, since the 2012 discovery of CSF1R gene, we've had a clinical trial, so we're moving pretty fast. I do have some disclosures, including that I'm a consultant and site PI for Visual Neuroscience who ran one of these trials, as well as Savannah Bio, which was formerly Novoglia. And today I'm just going to review what is ALSP, the diagnosis and management for CSF1R ALSP. We can be more specific now. And then the therapeutic development. So ALSP is adult onset leukoencephalopathy with axonal spheroids and pigmented glia. This was an autopsy-based diagnosis. You can see here from a white matter lesion at autopsy that within the lesion there's loss of myelin, there's axonal spheroids, there are some but reduced macrophages and microglia, and then everything looks sick, including the astrocytes and the neurons that are in the regions. It went by many names over the course of time until 2012 when Dr. Zolik's group from Mayo Clinic Jacksonville discovered that autosomal dominant pathogenic variants in CSF1R cause at least a subset of these ALSP cases. And so we could bring a genetic diagnosis to the clinic. There are CSF1R negative ALSP cases. Some of them are due to RS2 variants, which I won't talk about today, and some of them remain persistently undiagnosed. It is a rare autosomal dominant adult onset disorder. A recent study done by David Lynch's group looking at whole exome sequencing data from the UK Biobank found that there were 1 in 3,500 individuals with a pathogenic or likely pathogenic variant. They did not have a diagnosis of ALSP, but they did have diagnoses that can be misdiagnosed as ALSP, like Parkinson's, and many mood disorders were noted in the charts. So there's actually more than one CSF1R related disorder, and we don't yet know if this is a spectrum of the same thing or different disorders. But what I'm primarily talking about today is the autosomal dominant pathogenic variant that I told you was discovered in 2012. But there's also this more ultra rare, with 15 people reported in the literature, of this Bandos variant, which is brain abnormalities, neurodegeneration, and dysosteosclerosis. So they have bony abnormalities as well, and typically have earlier onset, and it's due to biallelic variants. The full knockout of CSF1R leads to early lethality, but there are people that don't have the full knockout and survive. And so far, but it may be because they weren't examined, the parents do not seem to be affected. This is a slide that I borrowed from Sisters Hope Foundation, the advocacy group for ALSP, to show that people all over the world have been diagnosed with this disorder and are willing to undergo treatment. There is a variable clinical presentation, which has led to misdiagnosis. So individuals typically present in the 4th or 5th decade of life. We don't yet know why it presents so late. The symptoms vary. It can be behavioral or cognitive changes. It can look like frontotemporal dementia. It can be quite insidious. I've seen people who initially presented or were noticed to have abnormal Facebook posts, for example, or sending nonsensical text messages to their kids. Then there can also be progressive motor impairment. It can be extrapyramidal, looking like Parkinson's, pyramidal, looking like MS. Women can present earlier in this cohort of 100 patients that have been previously reported with more motor symptoms. There's also sometimes seizures, cerebellar ataxia can vary between family members. And the MRI findings can be confused with other disorders like strokes or MS. And so because of this variability, there's a high likelihood of misdiagnosis. There's typically a diagnostic odyssey, and it can be rapidly progressive to death within 5 to 7 years after onset of symptoms. And so let's take a look at the imaging. So here's an example of the patchy paraventricular white matter that can show up, and this is what will get misdiagnosed as MS. There can also be extensive confluent leukoencephalopathy and atrophy, and typically corpus callosal thinning. However, I just want to note that the phenotype doesn't necessarily correspond to the severity of the MRI findings. For example, I have seen people who have the look like MS MRI that died within a year and were devastated. And I have seen people who had this extensive involvement and minimal symptoms. And so we haven't quite figured this out yet. There's the part that looks like strokes are this diffusion restriction. What's unusual and sets it apart is persistent diffusion restriction on longitudinal imaging. So it's really key to do that in people who are having these atypical presentations. And then on head CT, there is calcifications. So CSF1R is colony stimulating factor receptor type 1. It is required for the signaling for CSF1R is required for the survival and proliferation of the myeloid lineage cells, including microglia, the innate immune cells of the CNS. This is the first described microgliopathy. Shown on the bottom left is the dimer that forms between two CSF1Rs, and they respond to ligand CSF1 or IL-34 to lead to downstream signaling. Most of the ALSP pathogenic variants are in this intracellular downstream tyrosine kinase domain. So it's really not clear how these variants cause an adult onset autosomal dominant disease, and how does it lead to microglial dysfunction. So the options that are debated within the field are, is this due to haploinsufficiency or dominant negative or gain of function? And so I tried to make a picture to represent all the possibilities of haploinsufficiency, where you just don't get the pathogenic variant involved in the formation of the dimers. Different autophosphorylation assays have shown that, that this is impaired, leading to impaired downstream signaling. So that's at least part of it, but it's really not clear if there's additional dominant gain of function. And there's some data both for and against this. And so overall, this is a devastating disorder potentially without a disease-modifying treatment. And so the current standard of care is limited to symptomatic and supportive treatment. However, there's many current efforts to be clinical trial ready for disease-modifying therapy. We have a Sisters Hope Foundation, which is specifically for this disorder, and we have a Medical and Science Advisory Council filled with many enthusiastic members, which is wonderful. And we're working on developing consensus guidelines. We intend to develop consensus guidelines on diagnostic criteria and other clinical management guidelines, such as what to do with pre-symptomatic people and how often to monitor them. In 2022, we were incorporated into the Global Leukodystrophy Initiative Clinical Trials Network for the first time as one of the first adult onset disorders. And we met again in 2025. People that came to the meeting are shown on the bottom right. So to sort of push ahead, and we've actually made incredible strides just in those 3 years of meeting to identify the unmet needs for clinical trial readiness. This past year, we had an FDA listening session, as well as last month, an externally led patient-focused drug development session. And then we've actually had the Illuminate observational perspective study. It was terminated early because the company running it was purchased, and they are not purchasing the CSF1R aspect of their studies. However, this will have an upcoming manuscript, and we have learned quite a bit about outcomes based on what we did in the study. And I have started through the GLIA-CTN, what is currently a retrospective natural history study on CSF1R ALSP, and will at some point transition to a prospective study. And just to point out there, I put this chart up here, but got confirmation from Omar that we actually have 43 subjects enrolled now. And I just wanted to highlight Dr. Naidovich, who presented the work on developing this natural history study at ACTRIM so that we can educate our MS colleagues on finding this disorder so that we can diagnose it and help people learn more about the presentation and when to intervene. And Dr. Naidovich is actually one of my clinical MS fellows who's interested in joining me in clinic, so we'll see if that works out. So potential disease-modifying treatments. So allogeneic hematopoietic stem cell transplant has been performed in some patients. And so we have increasing data that suggests that it will stabilize disease. So there have been about 15 people in the US and a report in France combined into this number with the trend towards stabilization of the disease in the people that survived this intervention. There's some suggestion from these studies that intervening earlier with more motor predominant symptoms will have better outcomes. We just last week, there's a big splash paper in Science on ALSP and new mouse models and standard HSCT. And they report 8 patients, and they also reported stabilization. And there is a study that's coming out from the cohort in Europe, but we don't have that data yet to share. As mentioned, I think yesterday about BNT, we're not exactly sure what the mechanism is. It's proposed to be the replacement of the lost microglia because CSF1R signaling is required for the survival of these cells. They are reduced, so there is a niche for potentially monocytes and macrophages to land in there and start contributing microglia-like functions, but that has not been totally demonstrated yet. And another question for the even more rare variant of this disease is should people, including children with the Bandos variant, get bone marrow transplant? We don't know the answer to that yet. And then the treatment arm of the terminated studies was this phase 2 proof of concept clinical trial called the IGNITE trial that was also ended with the purchase of the company. It was trialing monthly infusions of a monoclonal antibody that was a TREM2 agonist called iluzanebart. And based on the press release, it had favorable safety, tolerability, and pharmacokinetic profile, but showed no beneficial effects on any biomarker or clinical endpoints, unfortunately. So obviously everyone is extremely disappointed in this outcome. This will be presented at a poster at ANA this fall, so I will be able to share more of that information on the outcomes at that time. And what I think is actually a huge success is that in this time, I diagnosed my first patient in 2019 that we've already run a clinical trial and had an outcome and learned and identified all the centers. So there are some successes, even though it was obviously a disappointing outcome. We have a lot of exciting treatments in development, including autologous HSCT with gene editing of the CSF1R variants. And I had permission from Yetta Lee and Jeff Gelfand to share. They just presented the work that they're doing at our ALSP community conference last month. And then there's also in the works this cell-based microglial replacement therapy, which if we're able to do it in this disorder would be a first in human. And is probably a good population to start it off in. Just in the schematic showing that they made iPSCs from patients with CSF1R, CRISPR corrected them, turned them into microglia, and then injected them into one of the mouse models called the fire mice, which do not have microglia to begin with, and showed some correction of some of the pathology there. So to be determined, but potentially very exciting. So obviously one of the major limitations we have is getting people to have a genetic test to show that they have this disorder. It's probably underdiagnosed based on the prevalence data. You need a multidisciplinary team care approach, and I'll talk a little bit about the challenges of that later. That we have advocacy support and research community. We have natural history studies. We have international collaborations. We have partnered with industry and developing therapeutic interventions. And this paper that came out in Science also has 2 new mouse models that seems to recapitulate the disease better than the prior mouse models. So we're definitely moving forward quickly. And so we're moving towards disease modifying therapy. And again, this is our grading system. We don't have it yet, but we're definitely moving towards it fairly quickly. So to summarize, there's many potential treatments in the pipeline. There's often delays to diagnosis due to the heterogeneity of onset and types of symptoms. The natural history studies will incorporate data from multiple centers to guide the future treatment trials. The community has successfully enrolled and completed a clinical trial, and ongoing studies on the underlying pathogenesis will influence the types of therapeutic interventions. Can we prevent this from happening? We have this advantage in an adult onset disorder to entirely prevent it, or can we correct and reverse it? So with that, I'll end there. Thank you.